So it's a real pleasure to introduce today Tomas Lindahl, and of course it's a real honor. Let me say that whatever I'm going to say about him is going to be an understatement. Tomas Lindahl has been working for many, many years on the mechanism of DNA repair, and also he was the first one to show that DNA is an unstable molecule. The implication of that discovery is huge. Think about the SARS devote lots of efforts in trying to maintain the integrity of the chromosomes and the DNA, and particularly to achieve promptly and with the right repair pathways the repair of the DNA lesions every time that happens. Think about that now our cells accumulate DNA damage, and luckily enough our cells are able to fix the problem, repairing the damage. And also cells have to choose the right repair pathway for the right cell cycle stage, coordinating all these processes, which is very, very important. Now, the relevance of this can be appreciated if you think about a failure to do this would inevitably result in cancer, because cancer is caused by mutations, okay? But also aging. The aging process is also caused by defects in DNA repair. With age, DNA repair goes down, and lots of DNA repair syndromes cause premature aging. Also neurodegeneration. Think about our neurons. They have to stay with the same DNA for 100 years and maintain the integrity of this molecule. So because of this discovery, Tomas has been awarded the Nobel Prize in 2015. And we were expecting for the moment. And I have to say that besides being a fantastic scientist, Tomas has been as an amazing scientific director, directed for many, many years a glorious center in Crick, the Crick Laboratories of Kansas City, UK, before SRF. And that place has been a place full of heroes for people like me working on DNA repair. Every time you go to a meeting on chromosome replication, recombination, DNA repair, you will meet people from Crick. And that's because Tomas has been directing so successfully that place. Okay, thank you very much, Tomas, for being here.
Thank you, Marco, for your kind introduction. It's a pleasure to be here in Bangalore, and I should start by apologizing to Marco and Francesco, my colleagues from Milano, who already have heard this story probably a number of times, because this is a long-term research project. And what it involves is the fact that DNA is not made of stainless steel and will ultimately decompose like all biological macromolecules do. So if you look at the first slide, please. This is a space filling model of the double helix of DNA. You have probably seen it before, and it looks very solid. Geneticists, of course, have told you for years that this is how inheritance is retained. So obviously we have to have very stable molecules to deal with our inheritance between generations. And the problem that comes up is that DNA as a macromolecule is composed of building stones that are not that stable. But if you look at the structure, it looks like a finished product. You have to be aware of that this is just the DNA part of the chromatin. And not even that, because DNA at neutral pH is a salt. So for each phosphate, which are the bigger of the components you see here on the outside of the double helix, there are neutralizing metal ions, potassium, sodium, magnesium, calcium, and so on. And They are important from the point of view that they preserve the double helical structure. If there are no counter ions, the two strands of DNA would fall apart. So if you're an experimental scientist, it's a very bad idea to dissolve DNA in distilled water. You always have to have some salts around to neutralize the phosphates. So that's one aspect that is not shown here, all the little counter ions that neutralize the charges in DNA. Another aspect that is not shown in this picture is water. DNA is a hydrated macromolecule. It has a big water shell around it. Much of that water shell you can remove just by replacing it with another solvent. And if you choose a solvent that's not corrosive or damaging, one can do such experiments. In our work, we have often used glycol, ethylene glycol, which may be familiar to you as antifreeze in your car, because glycol is a liquid that has a lower freezing point than water. And it's possible to make a solution of DNA in glycol instead of water. Such solutions at room temperature will retain the double helical native structure of DNA, with the obvious assumption there had to be salt there, so one has to have a glycol solution with salt in it, just like you have to have a buffer with salt components to dissolve the native structure of DNA. Well, what other roles does water play in DNA? We don't quite know, and there is water in the molecule that's essential, and we don't know what these are. The experiment one can do here is to dissolve DNA in glycol instead of water. To be more precise, that is 99.9% glycol. That works fine. But if you remove the last 0.1% of water from the system, the DNA structure collapses. Because within the double helix, there is a small number of water molecules per helical turn that are essential parts of the DNA structure. We don't know what they are. So we can't even show it in the space filling model. Where is the water in DNA? But there are at least a couple of water molecules per turn that are essential to retain the DNA structure. So there are still things we don't know about DNA structure, although we understand the genetic implications rather well these days. The problem I was interested in quite a few years ago is how can DNA be so stable when it's composed of simple chemicals, nucleosides, which are by themselves not that stable. Is there something other stabilizing factor? That seemed very unlikely, because these are simple rules of chemistry. If a nucleoside is labile, if you polymerize it into DNA, it will remain labile in many aspects. So if that is so, you would expect DNA to be so fragile that it shouldn't be able to exist as carrier of genetic information. That was the dilemma we addressed. And the answer to that question turns out to be DNA repair. DNA falls apart, but there are several DNA repair enzymes that specialize in repairing it all the time. So if you look at the next slide, shows one strand of the DNA double helix with 4 bases, A, G, C, and T. And what I've done in this picture is put little arrows in for the sites that are known to be of some chemical lability. You may have seen this picture because my colleague Bruce Alberts has written an outstanding and widely seen textbook in molecular biology and biochemistry, and he included in at least several issues these pictures. So what you see here are points of DNA damage, and I won't go through this big catalog. Point out that the most labile site in DNA are these base sugar bonds here, especially with purines. So this is a labile bond, and also you have the corresponding bond down here. This is the weak point in the DNA structure. Another weak point in DNA structure, which is of biological interest, is the amino group in cytosine, which is easily lost. And then the cytosine is converted to the base uracil, which has different coding properties. So that's potentially a mutagenic change in DNA, unless one corrects it somehow. So, and then there are other things going on. Now, if you lose a base here, you would lose genetic information, so you can't just ignore the change. So you have to correct it somehow. And its simplest would seem to be to just have an enzyme put the missing base back again. But that can't be done, because if you lose a purine base from DNA, it can be this one or this one, and the remaining structure, a sugar phosphate in DNA, is identical. There's no way of saying, just from looking at that sugar phosphate structure, if there used to be an adenine or guanine or something else there. So you have to use the information in the opposite strand of DNA to produce a more elaborate repair structure strategy, and that's DNA repair. So the first question then for a biochemist that comes up is, how common are these changes? And what we did was grow bacterial mutants labeled specifically in one or the other base in DNA. And you can do this most easily by working with microbial DNA, E. coli DNA, where you use E. coli mutants with defects in the nucleotide metabolism, so they are dependent on external addition of bases like adenine or uracil or cytosine and so on. That way you can label the DNA specifically in one component only of DNA and follow what happens with that. So that we did, and then we prepared such labeled DNA and studied its stability in conditions that are similar to those occurring in life, neutral pH. And you can't do this so easily at 37 degrees because the reactions are slow. But the DNA molecule itself is stable in buffer solutions up to about 85 degrees. So you can study DNA at temperatures like 65 degrees and 70 degrees and so on, and see what happens then. And then you will notice that DNA decomposes at a measurable rate. And we also know that the DNA structure at 65, 70 degrees is exactly the same as the one we have at 37 degrees, because there are thermophilic bacteria that will happily grow at 65 degrees. And they have the same DNA as we have. So by doing this kind of measurements over a period of time and so on. Next slide, please. We came up with some measurements due to computer hiccup that 900 should be here instead. But it tells you that 9,000 it is. It tells you that this is what happens in one genome in 24 hours at 37 degrees under in physiological conditions. So you lose many thousands of purine bases, and you also have other changes. These are experimental studies done with 100% double-stranded DNA. So some of these numbers actually turn out to be too low, because in some cases the double helical structure of DNA protects the components of DNA. So you say, well, that's fine, DNA is double-stranded, but you can't keep it double-stranded all the time if you want to replicate it. When you replicate the DNA, you have to separate the two strands. So there is a short period when the single strands are exposed. And during that time, some events, for example, deamination of cytosine to uracil DNA goes up over 100-fold, so it becomes a quite severe form of DNA damage occurring as a consequence of separating the strands during transcription and replication. And there are various forms of damage, and I'll come back to that a little bit. Much of the damage you see in DNA just in solution is due to hydrolytic cleavage or damage to the bases. Other forms of damage that are important are due to the fact that our cells get exposed to active oxygen during metabolism and our breathing. And that creates other changes in the DNA structure, which again are significant. And then there are several other things happening to DNA in cells, reaction with small molecules in cells. And I'll say a little bit about each of these, because none of them can be ignored. S-adenosylmethionine is the coin in transmethylation reactions, and it's needed in several metabolic processes in cells, and it also happens to be a reactive molecule. So next slide, please. Sequence specificity doesn't really occur for these slow reactions. There is a small exception which I came to light recently, so I thought I should mention it, that in the beta-globin gene there is one site which mutates about 5 times more rapidly than the average of all the sites in beta-globin, and that turns out to be, as shown by Fresco and co-workers, a site where there is a repeated DNA, inverted repeated DNA structure. So the DNA by breathing can form a hairpin structure, and that becomes a target for mutagenic events. It's a relatively small effect. If you add up all these mutations here, that's a much higher value than this single hotspot of mutagenesis. But it's still interesting that there are occasional rare sites in DNA where mutation frequency is increased. That is not true for most genes, but for beta-globin it is. So next slide, please. You obviously have to do something about this problem. You can't have tens of thousands of changes to your DNA structure and loss of genetic information. Life just wouldn't be possible under those conditions. And what a cell does is the obvious solution is that you repair the DNA all the time. So I mentioned that loss of a purine base in DNA is a damaging event. And the way you can repair this is use the information in the opposite strand to correct the error in a process that's now called base excision repair. Removes if a base has been removed, there's a special enzyme in the cell called NAP for apurinic sites, which this enzyme, which is abundant, doesn't cleave intact DNA, but it causes the chain break at sites where a base has been lost. And then there is a special trimming enzyme that can remove the sugar phosphate at the end here, so you create a little gap of one nucleotide in DNA. And that gap can then be filled in by a DNA polymerase. And finally, the DNA chain is rejoined by a DNA ligase, so you are back to the intact DNA structure, which was started with before it was depurinated up here. In mammalian cells, this is a distinct enzyme that doesn't have any other known function than dealing with these abasic cells. And this enzyme and this enzyme are all contained in the same polypeptide, which is DNA polymerase beta, which is a DNA repair enzyme that not only fills in gaps in DNA, but can also remove sugar phosphates from DNA. All right. One prediction of this model is that if this depurination happens all the time, you would assume that all these factors would be essential. Well, DNA ligases and DNA polymerases are also needed for other events, but this particular enzyme only is involved, as far as we know, in this kind of pathway. If you try to make a knockout mouse without this AP endonuclease, that is not possible. It's a lethal mutation, and that's what you would predict. If you can't cleave DNA at abasic sites, the DNA will accumulate abasic sites, will be of inferior quality, and this will lead to so many mutations that it's not compatible with life. So this is one of the arguments for this pathway actually being quite important. So I can move on to the next slide and show you what biochemists like to do, is identify components of pathways and reconstitute them. And this we have done here. So this is the pathway I mentioned with an AP endonuclease and then the rest of the base excision repair process. In some cases, if there is a damaged base, you need an additional enzyme. For example, if you have a deaminated cytosine in DNA, you first have to remove that deaminated cytosine. And this turned out not to be done by another nuclease, but a different kind of enzyme, which I call the DNA glycosylase, because it cleaves glycosyl bonds, not phosphodiester bonds in DNA. In other words, it's not a nuclease. It's a different type of enzyme, but that degrades DNA in a different way. And what it does is recognizes deaminated cytosine, which is uracil, and just cuts the base sugar bond. That doesn't cause a chain break in DNA, but it creates an abasic information-less site. And then the other enzyme, which I already mentioned, AP endonuclease, comes in and cuts the DNA at that site. And we can reconstitute this with synthetic oligonucleotides, which is shown here with the uracil in the middle. And what's not shown here is that it has a complementary strand, so it's a piece of double-stranded DNA. And one can then incubate them, such a construct with the uracil in the middle, with these different enzymes stepwise, And what you will find then is that what I've already said, that the DNA glycosylase doesn't cause chain breaks in DNA, but it sensitizes DNA to this enzyme, the AP endonuclease, which causes a chain break, and then the DNA polymerase beta in mammalian cells puts in one nucleotide in the little gap, and then finally DNA ligase repairs the DNA fragment here. So this is good biochemical evidence that what I'm saying is actually what is occurring. Next slide, please. You might wonder why DNA hasn't evolved to become more stable. The simple answer to that is that you can't make DNA that's more stable without losing replication fidelity. So it's possible these days to make various kinds of DNA analogs or homologs with other chemical compounds, variations on the usual nucleotides in DNA. And some of those are chemically more stable, but which is fine, but then when they are being replicated, they are not faithfully replicated. The DNA we have in our cells obviously has to be extremely carefully replicated. If you use various analogs to the building stones of DNA, you get sloppy replication and introduction of many mutations during the replication process, which is even worse than having label bases in DNA. So of the two evils here, it's better to have label DNA than DNA that can't be replicated very accurately. I put in something here that when DNA damage is a good thing, because in our cells, as you know, or our organisms, higher eukaryotes have antibodies which are essential to us if we want to live in a world which has many microorganisms and viruses in it. And there are many other compounds in the environment that are damaging to cells, so we have to have a large setup of antibodies, a big repertoire of antibodies to deal with all the insults that our cells can get exposed to. That would mean actually that a very large part of our genome would just be used to code for thousands or millions of different antibodies, and that's uneconomical. So what the cell has done here instead is introduce a hypermutation process, so that in the N-terminal region of antibody genes, those genes are particularly susceptible to an induced mutation process, so that one can change the structure of the antibody in response to an insult. And that's very convenient. So instead of having a million genes for antibodies, we get away with a few hundred genes of our antibodies, and each of those can be processed in response to an immune response. And there are two enzymes that are essential for this antibody diversity. One is the AID. It's a specific deaminase that can target this part of the antibody gene and specifically deaminate cytosins in this region. And through a series of steps I won't go into, it is possible then to process this change in different ways. The most common way is that you use the uracil DNA glycosylase to remove the deaminated structure. then you can ultimately get a variation, a broad possibility of changing the structure of these two genes so that you can respond to antigens in the environment. So this is an example of induced DNA damage, which is essential for our antibody response. Next slide, please. The kind of damage I've talked about so far has all been hydrolytic damage, water being involved in processing DNA in enzymatic processes sometimes. But as I already mentioned, there are other things that can damage DNA, like oxygen and reactive oxygen can cause damage to DNA, and we have characterized those different damages. There are several sites in DNA that are susceptible to active oxygen. They are almost as susceptible as sites for hydrolytic damage. And they are all different. So if you have found some process to deal with hydrolytic damage to DNA, it won't really help you to deal with the chemical changes that have been induced by oxygen in DNA, because those are different base changes in DNA. And there are a few of those, and one particularly interesting one, I think, is this one, which is 8-oxo-G, 8-hydroxyguanine. And this oxygen shouldn't be there. And if in oxidative process one has oxidized G that way, it changes the coding property of the guanine, so it no longer can be replicated in an accurate way. In fact, it tends to be paired with another purine base, adenine, and cause a transversion mutation. So this is a highly mutagenic change in DNA that the cell has to do something about. And the strategy here becomes the same as I've talked about when I talked about removing deaminate cytosine from DNA. That is, a DNA glycosylase recognizes this change in DNA and cuts out this oxidized form of guanine to generate a basic site in DNA, which can then be repaired, guided by the complementary DNA strand. Next slide, please. There are other things than water and oxygen that are dangerous to DNA. Well, some of the small molecules we have in our cells can interact with DNA and cause damage. And I've already, in passing, mentioned S-adenosylmethionine, which easily donates this methyl group here, and therefore is a cofactor in many transmethylation reactions. In fact, it's so reactive that it acts slowly by donating this methyl group even without any other provocation. So this is an alkylating agent. This is methylate, whatever it gets in contact with, which can be DNA or RNA or a protein. So there's a background of spontaneous methylation due to the liability of S-adenosylmethionine that happens all the time. And if you look at where those sites are in DNA, we find that they are at different sites from the kind of damage that we have induced with heat or with water or with oxygen. So again, we are back to square one. We have to think up some new strategy to deal with the damage caused by small molecules by acetylmethionine. So if you have that starting point, the first thing to find out is what does this do? And it turns out acetylmethionine causes exactly the same kind of DNA damage as simple alkylating agents like methylnitrosourea or MNG, which is sometimes used in laboratories as a mutagen-inducing compound. So all the time we have an alkylating agent in our cells, and there isn't much we can do about that. So there is spontaneous DNA damage due to alkylation. Well, what can the cell do about that? Well, again, I've already given you the answer. You have to repair the DNA. And here it turned out that there are a number of strategies that are interesting, at least to a biochemist, because they work by different mechanisms. Next slide, please. So there are at least 3 different ways you deal with alkylation damage to DNA due to this spontaneous methylation. One is that you first have to recognize what kind of damage is formed. And here we form 3-methyladenine. There's a methyl group here that shouldn't be there. Well, DNA can be methylated, and that's even used in epigenetics as a signal for methylation of DNA or for processing of DNA. But those methyl groups that are used in epigenetics are always located in the major groove of the DNA double helix. You can't have methyl groups in the minor groove of the DNA because that's where enzymes like RNA polymerase like to travel. So if you have a methyl group in the minor groove of DNA, it will block transcription and replication. In other words, it's a quite cytotoxic event. And what the cell can do about that? Same strategy again. You just cut out this space here by cleaving the base sugar bond and replace this toxic base with an unmethylated adenine molecule by the base excision repair process. And again, there is a separate DNA glycosylase that's doing this. By now, there are over 10 different DNA glycosylases known that remove specific forms of damage in DNA and screen for different forms of alteration in DNA. So now we have dealt with this one. There's an interesting variation here in that a rather unusual form of damage in DNA is O6-methylguanine, and that's an extremely mutagenic base, miscoding. So the cell, when it's formed, is very eager to remove it as quickly as possible. And the quickest way to remove this base is to have a repair enzyme that can recognize it and remove the erroneous methyl group here by a demethylation reaction. So this is a direct reversal of DNA damage. Doesn't involve excision repair of bases and nucleotides anymore. It just removes a methyl group that shouldn't be there. And that sounds like it would be, and it is, a very error-free and efficient way of removing this miscoding structure here. But there is a drawback with this. Now, when you move the methyl group away, it ends up on the protein that has removed it. So you now have a protein with a methylated residue in it. And surprisingly, it turned out that methylated residue is in methylcysteine. That was not known as a methyl group acceptor before. But you form methylcysteine in this protein. Okay, now to do more repair, you want to regenerate this protein and remove that methyl group. But you can't, because methylcysteine is an extremely stable compound from a chemical point of view. You can boil methylcysteine in hydrochloric acid and you still can't demethylate it. So at this point, the cell decides that instead of trying to break this extremely stable bond here, it's actually easier to make a whole new protein molecule. So this is an expensive form of DNA. You have to use up one repair protein, which is an enzyme, for each lesion that you repair. Another related form of repair is due to a couple of toxic lesions with methylated 3-methylcytosine and 1-methyladenine. Again, here are methyl groups in the minor groove of DNA, and they got to be removed because otherwise you have a toxic lesion. And we know that must be the case, and ultimately we found an enzyme in E. coli called ALK-B, which could remove this methyl group. It also has a homologue in mammalian human cells. And it took us some time to figure out how this works, because although we could take this ALK-B gene in E. coli and express it and make a lot of the protein and so on, when we mixed it with DNA, nothing happened. We just couldn't reconstitute a repair reaction, and the reason we couldn't is that there are a couple of unusual cofactors involved here, ferrocyan and alpha-ketoglutarate, which is an abundant small molecule in cells. Usually when biochemists try to do something, they don't add these components to their reaction mixtures, and then nothing happens. But if they are in there, you can actually reconstitute the reaction. And it turns out that this has become a fairly well-known strategy now for demethylation of also proteins. So you use, for example, to repair or alter methylated histones, to demethylate histones, you use the same cofactors, ferrocyans and ketoglutarate. So this is some of the things that happen just when you get exposed to one simple alkylating agent. Now, if you go back to your biochemistry textbook, you'll realize that a cell is full of reactive small molecules. What do the other ones do to DNA? We don't know yet. It seems quite likely that just like there are several DNA repair enzymes here that are necessary to deal with methylation damage to DNA, there are other forms of DNA damage being created, and that repair is going on all the time. But to define that, we first have to find out what that damage is like. Next slide, please. So this summarizes some of the intrinsic fragility data. There are group-specific reagents that can cause damage to DNA. Water. You may not think of water as a mutagen because we live in a water environment, but water is actually a weak group-specific reagent and is present all the time at a very high concentration. So this will cause damage to DNA and other molecules in cells. Slow damage, but with regard to DNA, something that cannot be ignored. Reactive oxygen I've already mentioned, and I said something about methionine. There are other small molecules in cells. Formaldehyde has been studied by K.J. Patel in Cambridge, UK, and that causes DNA damage. The form of damage has not been characterized in detail yet, but it's known that you have to remove alterations to DNA caused by formaldehyde damage. And as I mentioned, there are probably other things here, other small compounds that we should know about. Once we identify them, we can start looking for new DNA repair enzymes. If one finds new DNA repair enzymes, then you can then knock out those enzymes and see how they are related to DNA repair and even human disease. So there is a diversity of lesions due to the many different reactive compounds we are being exposed to, and this is just the beginning of the story here. And that predicts that there are other DNA repair enzymes we don't know about yet, because we first have to define the damage, endogenous damage that we deal with. Okay, and I have one more slide which shows some of the biological conclusions of all this. People ask, what are the mutagenic changes in DNA? We know today that the great majority of cancer is due to mutational changes in DNA, typically by inactivating so-called tumor suppressor genes, and this then causes an increased level of mutagenesis, which causes cancer. So what happens in cells? Well, we have known for a long time that radiation can damage DNA. UV light. UV light is an important component for understanding how skin cancer is induced, but probably not cancers in other organs. There is no obvious reason to believe that UV light is particularly important with regard to liver cancer, for example. So the skin is an important target, but otherwise UV is probably not explaining human cancer. Ionizing radiation, there are many scares about this. People are afraid of radiation, but the background radiation we are being exposed to is actually quite low, thanks to the Earth's atmosphere. It's a bigger problem for cosmonauts, or if people want to go to Mars, for example. This becomes a real threat to the genome. But otherwise, only a very small proportion of human cancer can be blamed on ionizing radiation, perhaps 1% or so. Food components is another common scare, but the food we eat is not really that mutagenic. We can easily deal with that. Because the Ames test and other tests allow screening, so we don't have to eat mutagenic food anymore. There are some parts of the world, let's say in Africa, if people eat very moldy peanuts because they're hungry, they will also consume a lot of aflatoxin, which is produced by the mold, and that is carcinogenic food. But there is no reason we should have food stuff like that to risk our lives. And in fact, I think the regulatory authorities are doing a good job of seeing to it that we don't have to eat mutagenic food. Smoking is a bigger problem, but people are aware of this now, that smoking cigarettes is a quite dangerous habit, and perhaps is slowly going down, and governments now start interfering and trying to discourage people from smoking cigarettes, which is a dangerous thing to do. Then there are other factors that can cause damage to DNA. I've talked a bit already about our metabolism and DNA replication. DNA replication is difficult to evaluate because we know that under normal conditions DNA replication is amazingly accurate. So there are very few mutations coming in due to DNA replication errors. But it to some extent depends on the counter ions in the DNA that I mentioned in the beginning. DNA where the phosphates are neutralized by magnesium or potassium is replicated faithfully. There's also some manganese in cells. A nucleotide in DNA with a manganese ion neutralizing the charge is more mutagenic by a factor of 10. And then there are other metals, ferrous ion, copper ion, which give mutagenic changes in DNA. So when we talk about how accurate DNA replication is, it's also very important to know what is the counter ion background that this has been done in. But all these changes, probably it's difficult to explain all the occurrence of DNA damage and spontaneous cancers from this and the remaining answer is this one, which has been something I've talked about. Endogenous DNA damage due to water and oxygen. You can't protect yourself from water and oxygen. Sorry. But it's always going to be there, and that means that we have endogenous DNA damage occurring, and there's not much we can do about that, and some of that will cause cancer. So when we talk about curing cancer, I don't think we will ever be able to cure all cancer. It will always remain there as an old age disease caused by components like this. So what we can concentrate on instead is finding better cures, better treatments. There is a certain reluctance to accept this as a major form of human cancer, because if you want to know what causes cancer, let's take smoking cigarettes, where the epidemiologists have done a great job in convincing most people by now that smoking is dangerous. Well, how is this done? What you do is you take two groups of people. One is heavy smokers, and the other group doesn't smoke at all. And then you compare them, and by standard epidemiology techniques, you will find that the people who smoke a lot have a much greater risk of lung cancer and a number of other unpleasant diseases. And that's an answer provided by epidemiologists. Now, if everybody was a smoker, we wouldn't know this, and we actually wouldn't know that smoking is dangerous. People probably would continue puffing away, and we would have a higher frequency of lung cancer than we have now, but there would still be people who occasionally would survive massive smoking. As you know, everybody seems to have heard of somebody's uncle who became 100 years old, although he smoked 3 packs of cigarettes a day. All you can say is he was very lucky. It's rare. But without a control group, you can't say that smoking is dangerous. And that's the problem we have here. I think biochemical evidence now suggests that compounds like water and reactive oxygen are quite dangerous to our DNA. There isn't much you can do about it except relying on repairing your DNA all the time. And this is what's going on. So I'll stop at that not very cheerful note. Thank you.